Hello and welcome to part 2 of our 4-lecture series on clinical genetic testing. Let's get started. In this lecture, we will dive deeper into the karyotype, chromosome microarray, and FISH methods of genetic testing. Let's start by breaking down the anatomy of a chromosome. Each chromosome has two main parts, the short arm, called the P-arm, and the long arm, or Q-arm. These arms are separated by the centromere, which is the region where the two chromatids, essentially the two identical halves of the chromosome, are joined together. The centromere is crucial because it's where the chromosome is pulled apart during cell division, ensuring that genetic material is properly distributed to each new cell. At the very tips of each chromosome are structures called telomeres. These act like protective caps, preventing the chromosome from deteriorating or merging with neighboring chromosomes. Telomeres also play a role in aging and cell health, since they shorten over time with each cell division. Understanding these key aspects is important because changes in any of these regions can lead to genetic conditions that we, as genetic counselors, often discuss with patients. Now that we've covered the basic anatomy of a chromosome, let's dive into how cytogeneticists use G-banding to assign numbers to different regions. Here on the slide, you can see a G-banded X chromosome. The staining creates a pattern of light and dark bands, which helps us break chromosomes into smaller identifiable regions. Numbering starts at the centromere and moves outward towards the tips, or telomeres, on both the P arm and the Q arm. Each arm is divided into regions, bands, and subbands based on how far the band is from the centromere. For example, the first band on the P arm is labeled P11, followed by P21, P22, and so on. Similarly, the bands on the Q arm start with Q11, moving to Q12, Q21, and beyond. The first digit represents the region, the second refers to the band within that region, and in some cases, there's a third level for subbands like Q21.3, which provides even greater detail. This numbering system allows geneticists to pinpoint exact locations on a chromosome. For instance, if a genetic test finds a deletion at Q or XQ21, we know it's on the long arm of the X chromosome in region 2, band 1. Understanding this is key when we discuss how structural variations impact health with patients and their families. This slide introduces some common cytogenetic nomenclature terms which help us describe different types of chromosome changes. First, a minus sign means that there's been a loss of a chromosome, while a plus sign indicates a gain. Del stands for a deletion, which means part of the chromosome is missing. DER is a derivative chromosome used when a chromosome has multiple structural changes or when it results from a translocation. DIC means a dicentric chromosome, which has two centromeres. DUP or dupe refers to a duplication, where a part of the chromosome has been copied. Isochromosome, or in parentheses I, means both arms in the chromosome are identical. INS is short for an insertion, where a piece of one chromosome is inserted into another. INV means an inversion, where a piece of a chromosome has been flipped around. .ISH refers to results from fluorescent in situ hybridization, or FISH, a test that helps identify specific DNA sequences or chromosomes. MAR, or MAR, means a marker chromosome, an extra piece of chromosome that can't be identified easily. R stands for a ring chromosome, where the chromosome forms a circular shape. T means a translocation, where parts of chromosomes have switched places. TER indicates the terminal end of a chromosome arm. Finally, tri, or TRI, stands for trisomy, which means there's an extra copy of a chromosome, as in Down syndrome, such as trisomy 21. Understanding these terms is important because they describe the nature of chromosomal changes we might find during genetic testing and how these changes could affect health and development. Let's talk about some types of chromosomal abnormalities. First, Robertsonian translocations occur when two chromosomes join together at the centromere. This often happens between acrocentric chromosomes, specifically chromosomes 13, 14, 15, 21, or 22. Individuals with a Robertsonian translocation can be healthy, but it may lead to fertility changes or an increased risk of having a child with a genetic condition. Next, reciprocal translocations happen when two different chromosomes swap pieces. These translocations can be either balanced or unbalanced, depending on whether any genetic material is gained or lost. In a balanced translocation, no genetic material is missing, so the individual may not have symptoms. However, fertility issues or pregnancy complications may still arise. Deletions occur when part of a chromosome is missing. If the deletion is large enough, it may be visible under a microscope. Smaller deletions, however, require more advanced testing methods like chromosomal microarray analysis, or CMA, which can detect subtle missing or extra chromosomal segments that traditional methods might miss. An additional type of chromosome abnormality is the formation of isochromosomes. In this case, a chromosome has two identical arms, usually due to the loss of one arm and duplication of the other. The most common isochromosome involves the X chromosome, but acrocentric chromosomes 13, 14, 15, 21, and 22 are also common candidates for isochromosome formation. These chromosomal abnormalities can result in a wide range of effects, from no symptoms at all to significant health changes. That's why genetic testing is essential for diagnosis and planning, ensuring patients and families have the support they need. Now that we are more familiar with chromosomes, let's take a deeper dive into the karyotype. This is an image of a euploid female karyotype, which shows a complete set of chromosomes from a female cell. Euploid refers to a cell that has the expected number of chromosomes for a species. In humans, this means 46 chromosomes, 22 pairs of autosomes, and 2 sex chromosomes, XX in this case. You'll notice each chromosome has distinct dark and light bands. Again, these banding patterns help identify specific regions on the chromosomes. Each band reflects areas of DNA that are either more tightly or loosely packed, aiding in the detection of structural changes like deletions or duplications. In the past, creating a karyotype was a very hands-on process. After staining the chromosomes to highlight these banding patterns, scientists would physically cut out pictures of each chromosome for a photograph, arrange them by size, and compare them manually. It was a painstaking task, requiring extreme precision and attention to detail. With advances in technology, however, we now use digital imaging and specialized software to analyze and organize chromosomes. This allows us to produce clearer, more accurate karyotypes more efficiently, helping us detect variations in chromosome number, such as aneuploidies, which are seen in conditions like Down syndrome or Turner syndrome. In this slide, we'll cover how to interpret cytogenetic nomenclature for karyotypes. Karyotypes are written using a standard format that provides important information about a person's chromosomes. First, we see the modal number of chromosomes, followed by the sex chromosomes, and then any specific chromosomal changes noted using cytogenetic abbreviations. The modal number refers to the total number of chromosomes in the karyotype, which is typically 46 for humans. This number includes both the autosomes and the sex chromosomes. Let's break down a few examples. For a typical female karyotype, we have 46XX. This means there are 46 chromosomes and 2 X chromosomes, indicating a female. Similarly, for a typical male karyotype, 46XY. Here, 46 chromosomes are present, and the X and Y chromosomes indicate male. Now let's look at some examples with abnormalities. We have 47XY plus 21. This means there are 47 chromosomes instead of the usual 46. The XY tells us it's a male, and the plus 21 indicates an extra copy of chromosome 21. This is a karyotype consistent with Down syndrome. In the next example, the T indicates a translocation between chromosomes 1 and 2. The information in parentheses, P32, Q22, tells us the breakpoints. The short arm, P, of chromosome 1 at band 32, and the long arm, Q, of chromosome 2 at band 22. Translocations can have various effects depending on the genes involved. In the next example, we have a female with 45 chromosomes, which means there's one fewer than normal. ROB stands for a Robertsonian translocation, a specific type of translocation between chromosomes 14 and 15. The Q10 tells us that both breakpoints are at the centromere of these two chromosomes, indicating that this translocation involves whole arms being swapped. This type of translocation is often associated with reproductive issues, though the person may not have any physical symptoms. Understanding this notation helps us interpret a wide range of chromosomal abnormalities and guide potential treatments or further genetic counseling. Let's take a look at some examples of karyotypes where the patient has a chromosomal abnormality. For example, this karyotype shows an XY individual with trisomy 21. As you can see, the modal number is now 47 due to the extra copy of chromosome 21, and a plus 21 has been added to the end of the cytogenetic nomenclature. Here we have a female karyotype with a Robertsonian translocation involving chromosomes 14 and 15. In this karyotype, the modal number is 45 and the two chromosomes have fused together, leaving us with one less chromosome. Since the individual is female, we see two X chromosomes here, indicating 46XX, but due to the translocation, the final count is 45XX Rob 1415. In this karyotype, we observe a supernumerary chromosome, which is an extra chromosome beyond the typical count of 46. This additional chromosome can arise from a variety of genetic events, such as duplication or abnormalities during cell division. The presence of a single supernumerary chromosome can have different implications depending on its structure and genetic content. While some individuals may be asymptomatic and lead normal lives, others may experience developmental delays or specific health issues associated with the genes present on that extra chromosome. For example, in conditions like Ring 21 syndrome, the presence of an extra chromosome 21 can lead to a range of physical and cognitive challenges. Today, advancements in genomic technologies such as array comparative genomic hybridization allow for more precise detection and characterization of these supernumerary chromosomes compared to traditional karyotyping methods. Thus, being able to pick it out on a karyotype is essential for genetic counseling and helps guide decisions regarding diagnosis, management, and potential implications for family planning. Karyotyping offers several significant benefits in the field of genetics. It provides a comprehensive analysis that can identify chromosome abnormalities greater than 4 to 5 millibases long, including trisomies, monosomies, deletions, duplications, ring, marker, and supernumerary chromosomes, mosaicism, and balanced structural rearrangements, including translocations and inversions. While karyotyping is a powerful tool in genetic analysis, it does have its limitations. Notably, karyotyping cannot identify submicroscopic abnormalities, which are those changes smaller than 5 megabases. These subtle alterations often require more advanced techniques like array-CGH or next-generation sequencing for detection. It cannot identify loss of heterozygosity or absence of heterozygosity, which can indicate the loss of genetic diversity at specific loci that might be crucial for understanding certain genetic conditions. Along with that, we have homozygosity by descent, which is often seen in cases of consanguinity, where two related individuals have increased chances of sharing alleles, potentially leading to recessive disorders. It also can't identify deleted genes of interest that may be critical for diagnosis, but are just too small to be detected with traditional karyotyping methods. Next, it cannot identify specific breakpoints or base pair locations, which require more precise techniques to map the exact location of structural changes. Karyotyping also struggles with detecting disorders related to expanded repeats, such as those seen in Huntington's disease, where repeated sequences are often too small for karyotypes to resolve, or methylation changes, which can affect gene expression without altering the chromosomal structure itself. Also, we have uniparental disomy, where both copies of a chromosome come from one parent, potentially leading to genetic imbalances. Finally, it's not great at picking up single gene deletions, duplications, or mutations, which just may not produce visible changes in the overall chromosome structure, but can have significant implications for health. These limitations highlight the importance of using karyotyping in conjunction with other genetic testing methods for a more comprehensive understanding of a patient's genetic makeup. Despite these limitations, karyotyping remains a valuable tool in various clinical situations. It plays a crucial role in confirming prenatal screening results by providing essential information about potential chromosome abnormalities detected during early tests. In cases of isolated congenital anomalies, where a single abnormality is present, karyotyping helps determine if there is a chromosomal basis for the issue. It is also used when multiple congenital anomalies are observed, either prenatally or postnatally, as it can uncover underlying genetic causes that explain the range of observed anomalies. Karyotyping is important in evaluating developmental delays or intellectual disabilities, where chromosomal abnormalities may contribute to these conditions and inform management and support strategies. It is also useful for assessing dysmorphic features, physical traits that deviate from typical appearance and may correspond to specific genetic syndromes detectable through karyotyping. In cases of infertility, chromosomal factors may play a role in reproductive challenges, and karyotyping can be an important tool in uncovering these factors. Additionally, it helps individuals who have experienced multiple miscarriages by identifying any chromosomal abnormalities that might affect pregnancy outcomes. Karyotyping can also identify or confirm chromosomal positions of genomic gains detected through SNP arrays, helping to correlate structural changes with clinical features. It is essential in specialized testing situations, such as when chromosome instability syndromes like Robert syndrome or Fanconi anemia are suspected, where genetic instability is a concern. Furthermore, karyotyping is used in tumor testing as identifying chromosomal changes can guide treatment strategies, while comparative genomic hybridization has become the preferred method for most conditions due to its speed, accuracy, and cost-effectiveness. Karyotyping remains important in specific clinical contexts where its insights are unparalleled. Shifting gears, let's delve deeper into the next genetic testing technique, the chromosome microarray. A DNA microarray contains thousands of short DNA fragments, called probes, assembled on a solid surface like glass. Each probe, typically 25 to 80 base pairs long, targets clinically relevant regions of the genome. Since the human genome has about 3 billion base pairs, it isn't practical to include every sequence. Instead, only the most important segments linked to known genetic conditions are added to the array, taking advantage of redundancy in the genome. Microarray technology excels at detecting genetic changes too small for traditional karyotyping, which has a detection limit of 5 to 7 million base pairs. For example, 22q11.2 deletion syndrome involves a 1.5 to 3 megabase deletion on chromosome 22, and Prader-Willi or Angelman syndrome involves 4 to 6 megabase deletions on chromosome 15. Both are changes that require microarray analysis for accurate identification. The CMA process begins with collecting a sample, typically from blood or saliva. DNA is extracted from the cells, labeled with fluorescent dyes, and mixed with a reference DNA sample, which serves as a control. Both the patient and reference DNA are hybridized onto the microarray chip, where probes bind to matching regions of the genome. A scanner then detects the fluorescent signals from both the patient and reference DNA. By comparing the signal intensities, the analysis identifies any regions where genetic material is missing in the case of deletions, or duplicated in the case of duplications. Overall, CMA provides a more detailed view of the genome, giving clinicians the ability to detect genetic disorders with greater accuracy than traditional methods. Now let's take a closer look at chromosomal SNP microarray analysis and how it builds on traditional microarray technology. While both technologies offer powerful ways to detect genetic changes, SNP microarrays go a step further by providing even greater resolution and additional information. The key difference lies in the type of probes used. Traditional microarrays detect deletions and duplications by measuring signal intensity. However, SNP microarrays use single nucleotide polymorphism probes to offer genotype-level data. These SNP probes or SNP probes target variations at a single base pair, which allows the test to pick up more subtle changes such as uniparental disomy or regions of homozygosity, something traditional microarrays can't detect. SNP arrays are especially useful for detecting consanguinity, imprinting disorders, and other conditions where identifying stretches of identical DNA from both parents is critical. This makes SNP arrays more effective for identifying recessive disorders and mosaicism than standard arrays. The SNP array's ability to analyze both the copy number and genotype data provides a deeper understanding of the genome, which can help clinicians identify more nuanced genetic changes that might otherwise go unnoticed. The process of sample collection and preparation remains largely the same. Blood or saliva samples are collected, DNA is extracted and labeled, and both patient and control DNA are hybridized onto the chip. However, the data analysis from SNP or SNP microarrays is more complex since it requires interpreting both copy number changes and the underlying genotypes. In summary, while traditional microarrays are excellent for detecting structural changes like large deletions and duplications, SNP microarrays offer additional resolution and help uncover genotype-specific abnormalities, making them a powerful tool in clinical diagnostics. Here we have an example of SNP array results. This visualization shows how chromosomal data is mapped and analyzed. Let's walk through the key features of this result. On the x-axis, we have the chromosome, while the y-axis displays both the allele frequency and log-R ratio. The goal of this visualization is to identify where genetic material might be duplicated or missing. Looking at the allele frequency section, we notice something important. The data splits into two distinct lines for the Q arm of the chromosome, while the P arm maintains just one line. This pattern indicates that the Q arm has been duplicated, resulting in 3 copies of the Q arm, but only 2 copies of the P arm. We also see a corresponding shift in the log-R ratio, where the signal shifts upward towards 1. The log ratio is a key metric used in SNP microarrays to measure the relative quantity of DNA in a particular genomic region. It compares the observed intensity of the sample's DNA to a reference sample, with the ratio expressed on a logarithmic scale. A value of 0 on the log-R axis indicates that the amount of DNA in the sample matches the expected amount. In this case, there's no duplication or deletion. But when there is a duplication or extra genetic material, the log-R ratio shifts above 0, typically towards 1, showing that there is more DNA present in that region. Conversely, when there is a deletion or a loss of genetic material, the log-R ratio drops below 0, reflecting the reduced amount of DNA. Here we see that the shift confirms the duplication, or extra material, in the Q arm. This slide presents 4 examples of SNP array results, each demonstrating distinct patterns linked to specific genetic conditions. The first plot shows trisomy 13, where the allele frequency displays 2 distinct clusters, reflecting the presence of 3 copies of chromosome 13, 2 identical and 1 additional. Instead of a clean split into thirds, the pattern reflects some regions that remain heterozygous, while others are homozygous due to the extra chromosome. The log-R ratio shifts upward, indicating the increased DNA content. In the partial duplication of 19q plot, the 2 distinct lines near the end of the Q arm in the allele frequency graph suggest the presence of an extra copy of this region. In the log-R ratio plot, the elevated signal in that same region reflects the increased DNA quantity, further confirming the duplication. In the partial deletion of 4p plot, the missing space in the allele frequency suggests a loss of heterozygosity, meaning 1 or more alleles are absent in that region. This is a key indicator that part of the chromosome's P arm is missing. On the log-R ratio plot, the dip below 0 confirms the deletion, as it shows that the DNA quantity in that segment is lower than expected. Finally, the fourth plot illustrates uniparental disomy of chromosome 15. The allele frequency shows a homozygous pattern, meaning both copies of chromosome 15 are inherited from the same parent, while the log-R ratio remains close to 0, indicating no gain or loss of genetic material. The pattern suggests an unusual inheritance, typical of UPD. These plots emphasize how SNP arrays can effectively detect a range of genetic abnormalities, including aneuploidies, structural changes, and inheritance anomalies. In the following SNP microarray plots for mosaicism, we can see the complexity that arises when there are mixed populations of cells within an individual. In the cases of mosaic trisomy 21 and trisomy 14, the plots illustrate how SNP microarrays can detect these subtleties in genetic makeup. Mosaicism means that not all cells in the body have the same genetic constitution. Instead, some cells have an extra chromosome while others do not. This variation can result in unusual patterns in the allele frequency and log-R ratio plots. We may see multiple clusters in the allele frequency graph, indicating different cell populations that either have the typical 2 copies of a chromosome or an additional copy. The log-R ratio may fluctuate in accordance with the proportion of cells exhibiting trisomy versus those that are euploid. These plots highlight the utility of SNP microarrays in assessing not only complete chromosomal abnormalities, but also the nuanced genetic variations seen in mosaic conditions. By capturing these complex patterns, SNP microarrays can provide critical insights into the genetic landscape of individuals with mosaicism, enabling more accurate diagnoses and a better understanding of the condition's implications. Understanding chromosomal microarray nomenclature is important for interpreting results effectively. Let's break down how we typically see this nomenclature. For a typical female karyotype, it looks like this: ARR 1 through 22 X times 2. This means there are 2 copies of chromosomes 1 through 22 and 2 copies of the X chromosome. For a typical male karyotype, it appears as ARR 1 through 22 times 2 XY times 1. This indicates 2 copies of chromosomes 1 through 22 and 1 copy of both the X and Y chromosomes. The notation ARR stands for array, which signifies the use of microarray technology to analyze the genomic data. The notation ARR HD19 refers to the genome build, specifically the Human Genome Assembly version HD19. This ensures we're using the correct chromosomal locations based on the latest data. In this nomenclature, the X indicates how many copies of each chromosome are present. The notation, or in parentheses number dash number, shows specific band locations of interest where significant variations were found. For example, the notation 4Q32.2, Q35.1, in parentheses 163, 146, 681, 183, 022, 312 times 1 indicates a 19.8 megabase deletion on chromosome 4 between bands Q32.2 and Q35.1. Another example, ARR 8 times 3, 21 times 3, indicates a single copy gain of both chromosomes 8 and 21. The notation ARRX times 2, Y times 1, reveals a single copy gain of the X chromosome in a male, which corresponds to 47XXY, a sign of Klinefelter syndrome. When you see ARR 1-22 times 3, X times 2, Y times 1, this represents triploidy, specifically 69XXY. Additionally, this example we see here ending in HMZC indicates homozygosity at location 11p15, which is associated with Beckwith-Wiedemann syndrome. Finally, the last notation we see here points to a 3.3 megabase deletion on chromosome 9 between bands p23 and p22.3. Chromosomal microarray testing provides several significant benefits for understanding genetic conditions. First, CMA effectively identifies genomic gains and losses, revealing clinically relevant alterations in chromosome structure. These genomic gains and losses include things like copy number variants and duplications and deletions. CMA offers high resolution for identifying submicroscopic abnormalities ranging from 10 base pairs to less than 5 megabases, which is crucial for detecting smaller genetic changes with significant clinical implications. It can also identify unbalanced translocations and their breakpoints, providing critical insights into chromosomal rearrangements. Additionally, CMA easily detects aneuploidies, which are abnormalities in chromosome number, such as trisomy and monosomy, which can greatly impact health and development. It assesses loss of heterozygosity or absence of heterozygosity, which are important for understanding certain genetic disorders and cancer predispositions. CMA can also reveal homozygosity by descent, indicating inherited genetic conditions, especially in consanguineous cases. Moreover, CMA is sensitive enough to detect mosaicism, identifying genetic changes present in over 20% of cells, which provides a comprehensive view of the genetic landscape. Finally, when used alongside G-banding karyotyping, CMA enhances diagnostic capabilities by identifying amplified or deleted genes of interest. While chromosomal microarray testing has many benefits, it also has certain limitations that are essential to understand. CMA cannot identify balanced structural rearrangements. These include rearrangements that do not result in a net gain or loss of chromosomal material, such as balanced translocations, which may be clinically significant but can go undetected. Another limitation is that CMA is not effective in detecting cultured mosaicism when it occurs in less than 20% of the cells. This can be particularly challenging in conditions where the mosaicism is subtle, as it may not be captured during testing. CMA also falls short in pinpointing the exact chromosomal position or location of genomic gains. This limitation includes the inability to identify specific features associated with markers, rings, or certain translocations. Furthermore, CMA is unable to detect disorders associated with expanded repeats, like those involved in conditions such as Huntington's disease. Methylation disorders, which can play a significant role in conditions like Prader-Willi syndrome or Angelman syndrome, are also not identifiable through CMA. Lastly, CMA does not detect single gene mutations, deletions, or duplications. This means that for conditions primarily caused by specific genetic mutations, other testing methods like targeted sequencing may be necessary to obtain a complete genetic picture. Recognizing these limitations is vital for effectively interpreting CMA results and determining when additional testing may be warranted. There are specific situations where its use is particularly beneficial. First, CMA should be considered when there is a need to identify breakpoints in chromosomal abnormalities found through karyotyping. This helps provide more detailed information about the genetic rearrangements present. CMA is also valuable for assessing isolated congenital anomalies. This includes conditions that are either anatomical or early physiological, such as a cystic hygroma, which may indicate underlying genetic issues. In cases of multiple congenital anomalies, whether observed before or after birth, CMA can offer critical insights into potential genetic causes. For individuals presenting with developmental delays or intellectual disabilities, CMA can help uncover any chromosomal abnormalities that may be contributing to these challenges. It is also a valuable tool for evaluating individuals with autism. CMA can detect genetic variations that may correlate with autism, providing a more comprehensive understanding of each patient's unique profile. Dysmorphic features, which often suggest an underlying genetic disorder, can also warrant CMA testing to explore potential chromosomal causes. Additionally, a history of consanguinity should prompt consideration of CMA, as it increases the likelihood of autosomal recessive conditions. In these cases, CMA can help identify any inherited genetic variations. Lastly, trio testing, which involves testing the affected individual along with both parents, can be particularly effective in pinpointing de novo mutations or inherited variants, adding another layer of detail to the genetic assessment. Now, let's turn our attention to fluorescence in situ hybridization, or FISH. This technique plays a vital role in the field of cytogenetics and complements traditional karyotyping and chromosomal microarray testing. FISH is used to identify the presence or absence of specific DNA sequences on chromosomes, making it an essential tool for detecting chromosomal abnormalities. It involves the use of fluorescent probes that bind to particular sequences of DNA. When these probes are introduced to the cells, they hybridize or attach to their complementary sequences on the chromosomes. Under a fluorescent microscope, we can visualize these hybridized regions, allowing us to see specific genetic changes. This slide presents the results of FISH analysis focused on identifying a chromosomal trisomy. As you can see, we have two images, one depicting interphase FISH and the other showing metaphase FISH. Interphase FISH is performed on cells that are in the interphase stage of the cell cycle, which occurs approximately 24 to 28 hours after the cells have been cultured. In this image, we can observe the fluorescent signals from the probes bound to the chromosomes. This approach allows us to assess the genetic material without needing the cells to be in the dividing phase. Interphase FISH is particularly useful for quickly identifying trisomy, as it can reveal extra copies of specific chromosomes even when the chromosomes are not fully condensed. On the other hand, metaphase FISH is conducted on cells that have been cultured for 5 to 7 days to allow them to reach the metaphase stage, where chromosomes are fully condensed and easily visualized. The second image shows the characteristic X and Y chromosomes in metaphase, where you can clearly distinguish the additional chromosome associated with the trisomy. This provides a more detailed view of the chromosomal structure, allowing for precise identification of the trisomic condition. Both interphase and metaphase FISH play important roles in diagnosing trisomies, providing complementary information. Interphase fish offers a faster turnaround time, while metaphase fish provides a clearer representation of the chromosomes in their most compact form. This slide illustrates the results of a fluorescence in situ hybridization analysis for a deletion syndrome. Here we can see two types of probes being utilized, a critical region probe and a chromosome reporter probe. The critical region probe is specifically designed to target a known area of interest within the genome that is associated with a particular deletion syndrome. In this analysis, the critical region probe will bind to the DNA sequences located within the area, allowing us to assess whether these sequences are present or absent in the test sample. On the other hand, the chromosome reporter probe provides a reference for the chromosome structure. It binds to a specific region of the chromosome itself, helping to visualize the overall chromosomal architecture. This is essential for identifying any deletions or abnormalities by providing a baseline for comparison. During fish analysis, the areas where the probes have bound will emit fluorescent signals which can be viewed under a specialized microscope. In this case, if the critical region probe shows a lack of fluorescence in the area where we would expect it to bind, this indicates a deletion in that region. This combination of probes allows for precise localization and characterization of chromosomal abnormalities, which is critical for diagnosing and understanding deletion syndromes. By identifying specific deletions, we can gain insights into the associated phenotypes and guide patients' management effectively. In this slide, we will discuss the benefits and limitations of using FISH in genetic testing. One of the primary benefits of FISH is its ability to deliver rapid results, which is especially valuable in reproductive settings, allowing expectant parents to make timely decisions. FISH can readily detect and confirm common chromosomal abnormalities such as trisomies 13, 18, and 21, as well as abnormalities in the X and Y chromosomes and specific deletions like 1p36 and 22q11. FISH is also useful for diagnosing conditions such as Angelman syndrome and Prader-Willi syndrome. With a detection rate of 80 to 95%, it is a reliable tool for identifying these genetic issues. However, there are some limitations to consider. The accuracy of FISH relies on the availability of specific probes, which can restrict testing if the necessary probes are not commercially available. Additionally, since probes do not always span the entire chromosomal region of interest, some abnormalities may go undetected. In reproductive settings, FISH results are not always considered diagnostic, so further testing may be recommended before clinical decisions are made. While FISH offers quick results for common conditions, confirming rare diagnoses can take longer, which may frustrate patients waiting for answers. In some cases, such as with IVF, custom probes may be required, further complicating and extending the testing process. In this slide, we will discuss when it is appropriate to consider using FISH in clinical practice. First, FISH is an excellent option, again, when quick results are necessary, especially in reproductive settings, as rapid testing can help alleviate patient anxiety when they're awaiting critical information. Again, FISH is particularly useful for confirming microdeletions or duplications that have been identified on a chromosomal microarray. This is vital for providing definitive answers and guiding management strategies. Furthermore, FISH can also be considered for familial testing when there is a known genetic abnormality in a family. In these cases, a custom probe may need to be created to test family members for the specific genetic alteration. Finally, FISH is employed in oncology for tumor testing. It helps identify chromosomal abnormalities in cancer cells, providing essential information that can influence treatment options and prognostic evaluations. As this lecture finishes up, some applied questions that I want you guys to be considering are, what does a karyotype miss? How about a CMA? Why may you select FISH over a CMA or chromosomal microarray? Why a CMA over FISH? Why may you order both a karyotype and FISH on a fetal sample? And what is a risk associated with chromosome analysis, not expected with single gene testing? Thank you all for watching. The next lecture will focus on single gene testing, multi-gene panels, and whole exome and whole genome sequencing. See you next time.